Hello, Jesse.
Good morning.
Morning.
I will be on camera in a little bit, hopefully,
Okay.
but I'm here for the time being.
All right. Yeah. Uh, yeah. Well, I heard from Ash. She can't make it today, but she'll watch the recording. And I don't know if Anson is going to be here later. And I haven't heard from anyone else this week, but maybe they'll drop in.
Okay. Excuse me.
All right. So welcome to the meeting. We'll start now. This is the last meeting before Neuromatch. So this coming week, we're going to have Neuromatch conference. It's going to be a whole week because it's sort of in lieu of SFN, which got canceled this year. See how they'll pull it off. I don't know the details of how the talks will be recorded, but we'll see. I guess they usually do a pretty good job of handling a lot of the technical challenges. They're like probably about 1,000 or I don't remember how many talks there are. There are a lot of talks. I don't think 1,000. It's several hundred though. I saw the list. So let's see.
One thing about I don't know if you were gonna say this, but for anybody who's watching, there's a big need for volunteers, and especially for people to kind of help out. Even just kind of managing stuff for what they call showrunners or people who are going to be able to like the back end and not so like you can be like you can be a host for the many talks that are there, or like an MC, not a host, but like someone to facilitate them, or just helping out kind of on the back end technically, or arranging things or coordinating things. There's a big need for that. Coordinating's been talking about that a lot, and I'm in the volunteer Slack as well. And I may try to do some of that myself. Actually, I signed up for a few time slots, but I have plenty of things to do. But I'm going to help out when I can. But there's a big. If you have any free time, there's time slots available all over. Like it's a 24/7 event basically, for a week. So there's a lot of talk time, and a lot of need, and a lot of availability. So anybody who is free time and wants to help out, there's an Airtable. It'd be good if I retweeted it, but there's an Airtable link, and maybe the lab could, or I will put it in a weekly communication or something too. Just because there's a big need for that, yeah. Anson.
Anson, how are you?
Pretty okay. I stayed up a little bit last night, but actually not feeling groggy, so that's pretty good.
That's good.
I think it's the new CPAP mask I got to large now. The last one is medium.
Well, that's good.
That's good. I was just we were talking about Neuromatch coming up, and how are you guys doing on your presentation?
Okay, but I haven't really had anything together yet.
Okay.
So that'll be like today.
Yeah. I think you guys go Monday, so.
Yeah.
But it's okay. Well, we'll talk about that a little bit later on. So why don't I go to Neuromatch, actually, the website. We'll look at that a little bit, see how that's the upcoming events. So this is the Neuromatch 3.0 website. It says neuromatch.io, but they update it. So if you want to attend, of course, you can register until the end of the conference. Submissions gone. Registration fee is $25, but if you need it waived, you can have it waived. And then they have the different themes here from A to F. So we have papers or talks in most of these tracks. I think the only one we don't have toxin is paradoxically theme A, which is development, neurodegenerative disorders, and injury. The developmental AI talk is in this theme D, cognition, motivation, and emotion. And so that'll be an interesting time. The agenda, there's an abstract browser and an agenda. So why don't we look at the agenda? So this is the agenda. So it starts, this time is my time. So this is like 9 a.m. Eastern time in the US. And I don't know what the UTC time is. I think 1 in the afternoon. So this is Neuromatch 3.0 opening session, which is just kind of welcome to Neuromatch. Then they have some keynotes in the morning until 11 a.m., which would be like 3 hours after the start time. So this is when we start getting into our talks. So you can see that they have all these different like talks. I don't really know how this is organized, but this is your talk matrix. These are the traditional talks, I think. I mean, some might be interactive, but I'm not sure how this is. Basically, I think it'll be like keynote versus like some of the traditional and interactive. Oh, they're labeled. Okay. So these are the interactive talks in like blue or whatever this color is, and this is green. And so, a lot of interesting talks. There's a talk on connectomes, a talk on receptive field geometry, and so forth. There's a panel at 12 noon, panel on future opportunities in neuroscience with Brain Initiative Alliance. Then I think, there are more keynotes here. And then I think let's see, when is the talk? 4:30 is the trajectories talk. This is Anson and Jesse. So this is 4:30 my time, which is 5:30 Eastern time, which I think is the time zone for both of you. So that should be interesting. And then this goes on for days here. You can see like at the middle of the night in North America, there isn't much going on. But then at 3 a.m. it starts up again. And it goes down. I think our next talk is like Wednesday and Thursday. Those are the 2 days when we have talks. I'm going to show a spreadsheet in a little bit of our talks and the times. So we'll have that. You don't have to search through this matrix. But there's a lot of really good stuff here. I recommend that you look at all of this, all of these talks, and try to attend the ones that, you find interesting, because it really does stimulate your sort of how you think about problems, even if it isn't in your area. It can be very useful sometimes to see some of these other talks. So yeah, it's going to be really good. And then it'll go through Friday. So we're already on Thursday here. Goes through Friday evening, North America time. So there's it's at 7 p.m. Eastern time. There's a Neuromatch 3.0 closing session, which is the wrap of the Neuromatch. Now, in terms of like recordings for the sessions, I'm not really sure how they're going to handle it yet. I really hope that it all gets recorded. I know they were recording to Crowdcast, and then they switched to YouTube during NMA, but I don't know what they'll use this time. But hopefully they'll be recorded and available after the fact. So if you miss something, then you'll be able to go back and see what you missed. Now, I can't make your talk on Monday because I have a prior engagement. I'll try to make the end if I can. I'll try to make it if I can. I don't know how long that meeting I'm going to have will last, but that'll be, it'll be, hopefully I can catch it in recording. So this is the spreadsheet here. And let me, So this is the spreadsheet with all the times, and hopefully the sharing's on for that, with all the talks and the times. So we have, let's see, 6 talks. These are just all people in this group. So we have Psychophysics of Non-Neuronal Cognition. And this is from the Devorm group, but we have Ashk and Jesse on it. From this group, myself. So this is Thursday, and I have the times broken out here. So this is 12:30 UTC, which is 8:30 Eastern Standard Time, 7:30 Central Standard Time, or Daylight Time, I guess we're still in. So that's going to be pretty early. But, hopefully that gets recorded. And, of course, it's 6 p.m. India time, so it'll be in the evening there. Developmental AI is Thursday as well. This is the one with the one that most of us are on in this group. And this is the one, I'll show the talk in a minute, what it looks like. This is Thursday. This is in the afternoon on Thursday. So this is 2:30 Eastern Time, 1:30 Central Time, and then 12 a.m. India time. So if you're in India, some of these times might be inconvenient for you if you want to see the talk, if you want to attend the talk. So if you just watch the recording, that's fine. But, that's the way it goes in these conferences. If you have a distributed conference like this, if you have one time it's good for one time zone, it's bad for another time zone, and that's why they record them. So, trajectories of cognition, of course, that's Jesse and Anson. This is going to be Monday, the 26th. That'll be 4:30 p.m. Central Time, 5:30 Eastern Time. And then that's, of course, 4 a.m. India time, so that's pretty inconvenient for them. But I think that'll be recorded, and that'll be an interesting thing to watch. This is in the history and philosophy section. So definitely keep us posted on that. Convergence Insufficiency is a talk that Ashk submitted, and I'm on that with someone from her university as well. And that one is on Thursday, and that is very early for me, but it's 4:45 India time, so that's good for India. And then 10:15 UTC, so it's also good for Europe. This is the link to the slides. So last time I was in those slides, I didn't see a lot in there. They had the outline and the actually I did see a lot in there. It was just that it was an outline mainly, and then I did some work on some of the diagrams and things like that. So that's, I don't know if it'll open it for me. We might come back to that later. Then there's the BNN's ANN's talk. That was Thursday the 29th as well. That's at 1 p.m. Central Time, 2 p.m. Eastern Time, and 12:30 India time. And that's the talk I have with, it's me and Krishna Katyal from the Devorm group. And that's actually his time zone, so I don't know why it's so late. But let's see if I can open it up here. So this is again, I think he's in here, all right. So he's in here currently, so that's good. This is the talk, the title slide, and then we go through the comparison between the two types of networks. And then we have our outline here. What is a biological neural network? Here's an image of a motor neuron. Artificial neural networks, a definition of it. And then this is a slide that I was going to put in some information on, but I haven't done that yet. This is an example of a artificial neural network. Maybe this goes up here, okay. And then this is the difference between an ANN and an BNN. So this is, of course, then the basic difference. And this is in BNNs, you have neurons that fire asynchronously, parallelly, having dense neurons. Well, I'll have to change sort of the way this reads a bit, but I'll do it later when I'm not trying to talk about it. And so this talks about how they partially follow the power law. And I'm not sure if we should get into power laws here. Well, I think we should maybe add another slide on connectivity patterns, but that's because when you get into power laws, it's a little bit curious, but it also can be very messy because you can either gloss over it or go on about it for like 10 slides. So, yeah, that might be something we want to revisit. There's a theory of deep. This is another note I made. I haven't done this yet. Theory of deep networks, connectionist networks. Just kind of mentioning things about connectionism and about the between that and biological brains. Then there's a section on power consumption, so that's something that we'll probably talk about in this one. Fault tolerance and size, which is an interesting topic. And this is just about how, if you have a problem in the network, like, failure of the network or something like that, BNNs are better at fault and ambiguity tolerance. But there have actually been some papers on biological brain, like brain neural net or biological neural networks that have dealt with what they call adversarial attacks. So, like, they have, you have a brain network that's a bunch of neurons that are connected, and you can actually measure these in neuroimaging data. So you can measure different, you can treat different brain regions or neurons or areas of the brain as like a node, and then they're connected together. And then you can actually look at how those change when you introduce like cognitive perturbations, like a lapse of attention or some where you get people to forget about something. And so we might add some papers on that. And then that's the end of that talk. So that's like probably a good 15 minutes right there. So that's that talk. If you're interested in that, you'll be able to attend that. Hi, Krishna. How are you?
Hi, how are you?
Good. We were just talking about the presentation. This is the one that you and I are presenting. Is there anything you'd like to add about it, or?
No, I have nothing to add. Actually, I was trying to join the Google Meet link.
Oh, yeah.
So then I messaged you regarding that, and I saw the email and I came to know that it was on this seat. So apologies for, you can say late arrival.
Oh, yeah, no problem. It's we do a different meeting spot, but yeah, so we were just talking about Neuromatch and the schedule for the week. So this coming week is the whole conference, where this BNN's ANN's talk is Thursday. So I don't know if I sent you this link to the spreadsheet, but there's a spreadsheet. Yeah. Good. So that's that talk. And then there's also, this last one is Rashab and one of his collaborators. It's called The Mind's Eye. It's like a deep learning type approach or something like that. I can't remember right now, but I put that on the list so that we can remember to go attend it. This is Wednesday. This is 8:15, 9:15, so 9:15 Eastern Standard Time, 6:45. I think it's AM. I don't know why. I don't know why it's so early, so off time here for India, because I think they're in India as well. But that's the time that they're presenting. And again, if you want to find the exact name, it's on this provisional schedule. I think they're going to post a schedule with links to the sessions. So I think when we approach Monday, they'll have a new schedule with a list of sessions and the links to the sessions, so you can actually attend these, because I don't know where they're going to be hosted. That's what Jesse was talking about with the volunteers. I need volunteers to host talks and to be MCs for the sessions and that. So when we get more information about that, we can pass it on. I'm kind of looking for this Mind's Eye talk. I don't know if it's in here or not. I can easily pick it out. But there are a lot of really interesting talks in here. Some are pretty theoretical and some are not. Some are pretty detail-oriented, as you might put it. Like neuroscience needs philosophy to understand representations. It's really kind of an interesting topic, given what we're interested in this group. Let's see if I can find
When is that one? I missed that.
Oh, that's, I think, in 7:30 in the morning my time, so it's 8:30 in the morning your time.
Oh,
Let's see. Did I say 1:30 for, no, it was, yeah, I'm not thinking, let's see. Here it is. Oh, okay. I'm sorry. I have this timer. It's 8:15 p.m. for my time. Okay, that's what it is. Oh, I did have that right on here. Okay. So it's 8:15 p.m. This is the Mind's Eye, Seeing Through the Mind's Eye, Reconstruction of the Visual Stimuli using 3D Generative Adversarial Modeling. And then this is the abstract here. They have a self-supervised neural decoding pipeline. They're performing 3D reconstruction on 2D stimuli. So they're doing this encoding from 3D to fMRI and fMRI to 3D. So an encoder and a decoder architecture works. So they're doing all this stuff, and they're this is basically neural decoding for a brain-machine interface. So they're doing, I guess they're trying to use neuroimaging data to predict visual stimuli or things that you would see in the world. So it looks interesting. I don't know much more about it though. So, but those are the talks. So be sure to, if I think I have this link in the Slack. If you need the link, send me a message in Slack. If you have your slides and you want to link your slides in here, put the link in the shared slides area here. And that's the only link I don't have is the Trajectories of Cognition link. And the Mind's Eye link. So those talks can be put in there if you want. If you want me to review anything before your talk, send me the slides. I can take a look at them really quickly, make some suggestions. Actually, we can go through the other talk as well, the Convergence Insufficiency talk. Let me see if I can open it up. So this is with Ash and myself and someone else from her institution. Overcompensation of lens accommodation in the case of convergence insufficiency. So this is a theoretical model. There's this accommodation triad, which is sort of the model they're working from. This is an image of the eye relative to the brain. So you have this ocular motor system. You have, and this is kind of an outline of the work. This is a diagram of the interaction model of accommodation and vergence control. So it's like a control model. And then we go into just kind of compensation by vergence, what that means. This is how do you manage this clinically? How is this possible? Research scope. and then future directions, and this is an agent-based model slide that I put in, and then the references. So this is still a work in progress, but we'll get it finished by the time that it needs to be finished. And again, that's something that is also going to be given. So, any questions before we move on to some other business, or? Do you have any questions, Jesse or Anson or Krishna?
Not for me. But
Not me.
Okay.
Nothing from my side.
Okay, that's good. So why don't I go into the other two? These are the group talks. So I kind of made these slides from some of the slides that we've used previously for these projects. So let me go back to the schedule here. So these are the Psychophysics of Non-Neuronal Cognition and Developmental AI. So these are going to be on Thursday. And these are slides that I have links to them in the Slack. So if you, it's in the general channel. So if you want to look at these and suggest changes or additions or whatever, you can do it in that Slack channel at that link. Or you can go to this spreadsheet as well and click on the link to it. So this is approaching an artificial intelligence as an embodied developmental process. And so a number of people in the group are on this talk. And this is the sort of the intro slide where I have some GIFs of different embryos and developing nervous systems. So this is just a zebrafish embryo. So this is a very early stage where there's a lot of cell migration. There isn't really a brain yet. Then we move on to this. This is an axolotl nervous system. So this is a type of amphibian. And this is early on in development when the nervous system is just setting itself up. And you can see the sort of the way it's setting up here in the in utero stage. And then this is a zebrafish notochord, or elongation of the zebrafish notochord. So this is a little bit later in development from this example, where there's like a notochord forming and it's straightening out, and then the rest of the zebrafish will develop. So this is the idea behind sort of why in development may be important, because we're getting that formation of the morphology and initial wiring of things, and that's why we're interested in it. We can implement this in something called Bratenberg vehicles, which are an old model of sort of a toy model of cognition. And the thing is, it allows you to look at first order stimulus response mappings. It allows you to take stimuli in the external world and use an embodied nervous system or an embodied set of sensors and effectors to translate that stimulus. So this is just, you take an observation of the empirical world, you put it into this, what essentially is a black box in the classical case, and it gives you a behavioral output. And so they're very simple neural networks that are embodied agents. And so the bodies are very important because that's where the sensors and effectors are. But we can also think of this differently. We can think of this as being a developmental system. So in this case, we're not really evolving, or we're not really developing the bodies at all. The bodies remain constant. But inside the bodies are nervous systems that develop from simple sensor effector mappings to an actual network inside the body. And so this is what we talk about network morphogenesis. So network morphogenesis means that these networks inside sort of develop or grow over time. And so now you get, instead of a simple mapping between sensor and effector, you get a complex network of interneurons, and that attenuates the feed-forward signal. So this is an example of it, where you go from a simple mapping that I just showed you to these layers of nodes, and they are connected in different ways. And so there are 3 layers in this case between sensor and effector, and there are different types of connections between them. And then later on in development, you get a more complex set of connections with still only 3 layers, but there are more cells in each layer. So there are more nodes in each layer, and there are more connections between the layers and between different sort of pathways from sensor to effector. So this is the Chromotaxis. So this was a slide I had in the Devo Neural Networks talk for that workshop. And this is on something called Chromotaxis. So this is an example of a behavior generated by what we call metabrain model, which we can build using a DBV. And instead of going through what that is, I'll basically describe this phenomenon called chromotaxis. So if we think about like a taxis, it's a movement towards a stimulus. As a phototaxis, you have this simple attraction-repulsion mechanism. So you can be attracted to certain colors of light or certain colors on this gradient. As a preference, this can be a separate continuum emotional valence. So these different colors can be interpreted in terms of different emotional states. So if the vehicle is attracted to one state or another state, it might exhibit other behaviors consistent with some sort of emotional valence, or it could be interpreted as an emotional valence. It's hard to know with the whole language of Brainberg vehicles is sort of vague like that, but we can think of it that way as well. Then you can also look at the differences between congruent stimuli. So you can look at like the differences between these 2 stripes, and like the vehicle being able to distinguish between these 2 stripes. Something like this, which is also spatially distinct, enables what we call generalized Hebbian learning. And so if you want to read more about this, you can read this paper. Or we have another paper forthcoming that describes this a little bit more detail. And then you can also look at this in terms of a differentiation between incongruous colors. So like between this one at the left end and this one at the right end, these 2 stripes. These are incongruous colors. They don't blend as such as these 2 do. And you can use, this enables spatial learning. So you have 2 different colored stimuli that can enable some spatial learning mechanism. And this can be demonstrated using a genetic algorithm approach called ProgenBrain. So we've done a lot of prior work on this, and I use Chromotaxis as an example. But this is all enabled by something called connectivity activation and coding. And you can think of this as this WIJ matrix, and we'll refer to this in the next few slides. So we have a matrix that is this set of nodes and this set of connections, and W is the matrix, and IJ are the connections and their weights. So each developmental time point produces its own connectivity matrix. Each matrix has a corresponding determinant, so that determinant then represents the entirety of that matrix. But what is WIJ? WIJ is a summary of connections among a network of size K. But we know that in development, the number of neurons K can grow, and the number of synapses IJ can fluctuate in terms of their strength and efficacy. So WIJ is scaled by parameter K. So that means that you have matrices of different sizes. And so that's something that we have to think about during the development of these nervous systems. But we actually don't necessarily care about the WIJ over time. What we care about are universal functions that might result from this nervous system. So people have thought about this. There's been a paper on universality in neurodynamics across large populations of networks. And, that's maybe a way forward here in terms of thinking about how this developmental process produces, something that's coherent in terms of a network and how it behaves. We also kind of shy away. We think about development as sort of a reductionist representation, at least in the first slide. And that's if you add all components of a biological network together. That's, so if I go back to this first slide here, this is a reductionist representation of a developing nervous system. You have all these connections, you have, growth cones and things like that, synaptic sprouting, all these different processes going on. But in this case, we don't care about that so much. We just care about these, what look like neural networks. And so, what do we need to add to WIJ to make this model more biological? Or can we add biological realism without finding universal features or making the model more descriptive? So we don't, what do we need to add to WIJ to make it more biological, but biological in a way that's informative? We can add all these different features of development, but it doesn't necessarily mean it gives us anything that's, more informative than what we already have. So this set of slides here, I may have to work on the wording a bit because it's a bit clunky. So if you have suggestions for the wording here, and in this slide too, I wasn't particularly happy with the wording, but anyways, so this is an example of our matrix and our determinants over time. So this is using an example of C. elegans. So this is the adult C. elegans, and then these white dots here are the neurons that are born at different points in development. So these neurons are born in what is basically an oblong sphere, and they sort of move around a little. Well, they don't really move around too much, but the morphology sort of changes shape from that oblong sphere into this worm. And so these cells are born at a certain time, and they connect using gap junctions, and then they make connections using axons and synapses. And so there's an eventual connectivity of these cells. But you can see that you get cells that are born in different parts of the nervous system at different times. And so this matrix of cells changes over time. There are more cells and in different places. And this is mediating behavior. So by the time the worm is born and it starts to move around, it's not, necessarily just muscle twitching or cells moving around. It's this network actually activating. And so, we'll move next to critical periods. So critical periods are this window of developmental time that features an enhanced ability to learn. So we talked about the size of the matrix. Now we talk about this sort of rate of learning that is encoded in the developmental process. And this critical period is generally associated with physiological reorganization. And in our case, this is just this WIJ matrix, the weights in this matrix changing with what is in a biological organism, physiological reorganization. But in a network like this is just, changes in weights. So the critical period can have a variable width over developmental time. It can be shifted earlier or later in development. And so this is another concept that we use called developmental freedom. So the way to characterize this is to think about our developmental network in two modes. So in the first mode is morphogenesis, which is defined by slow growth before the critical period and fast growth during the critical period. So you have this nervous system that starts to grow and see an slow increase of neurons and connections. Then you have this critical period where there's an explosion of neurons and connections. And then you move into the next phase, which is developmental learning. And this phase is defined by no growth in the size of the matrix or relative stability among connections. But it doesn't enable this developmental freedom or the ability to learn on this now fixed or nearly fixed topology. And so that's what we have to remember, is that this network can grow, and then once it becomes sort of saturated with neurons and maybe with connections, then you can start to learn things. And then that in and of itself has, there's some change in the weights during the learning process, but at least you're not adding new connections or new cells to the mix. And so the critical period is managed not only by developmental freedom, but by contingency. There are 4 different types of contingency we looked at: an early critical period, a late critical period, early developmental freedom, and a late developmental freedom. And so if we go to A, there are a number of attributes. The adult neural network tends to be more homogeneous. They exhibit the same types of configurations and motifs. There's less time for preliminary or auxiliary nervous system or behavioral traits to develop. And there are fewer varieties of experience prior to maturity. A late critical period, by contrast, is an adult neural network exhibiting a larger number of possible configurations. Slower developmental periods afford more time for exploration and wandering. The early developmental freedom period is a low degree of overall developmental freedom. This early critical period allows for faster ascendance to a fully mature suite of behaviors. And late developmental freedom is slower ascendance to adult behaviors and capabilities. I think I'm going to have to, I wanted to put this into maybe like a contingency table. So I'm probably going to change this around, but that's basically where you're looking at critical period versus developmental freedom. And you have these 4 sort of categories. And, the problem with doing that is they're independent. So it's hard to say, but I can do this later. Just have like 4 boxes each with their own attributes. So these attributes, instead of being a wall of text, would be sort of, just spelled out. And then I don't know if we're going to have a lot more time after this, but there's the Gibsonian information. And fortunately, this section is very visual, so it won't take much time. But we think of the information in these networks as sort of Gibsonian in nature, which means that motion provides information. So we can think of things like optical flow or relative motion, and there's structure in motion that's used as information. So one of these is flow, motion, and contrast. So this GIF of a roller coaster is an example of visual flow, and the information that's embedded in this image, there's experiential information as well as spatial information and other types of information as well. Time information. Another thing we can measure is disjoint distributions. So this refers to moving versus stationary pixels. So you have this animated GIF of like a static or white noise. So if you watched an analog television, you know that this is a screen you can get where there's white noise static, and it's, this is just a random process underlying this. But you can have a single sort of screen capture of this, and you can compare the two over time. So this is an example of stationary pixels. This is an example of moving pixels. What information, if any, do you get from motion versus static representations like this? There's a time series structure as well, so there's temporal dependence. So if we have, this is the same little movie clip, but this one is missing frames, and so the action is in the same sequence, but it's missing frames. You can see that there's a asynchrony here between the two things. So can we sort of extract time series structure out of these and compare them? We can understand like scenes and things like that that the vehicle might observe in the real world in terms of this temporal dependence. And then finally, you have maximum versus minimum coherent movement. So this is a bunch of balls moving around stochastically in a box, meaning they're going at random speeds and directions, versus something like this where you have sort of a maximum coherent movement. It's a flock of boids, I think, which are digital organisms, and they're moving around in this concentrated way. And so you can compare these two different modes of movement, much as you can compare these two modes of video and extract information out of them. And this is this Gibsonian information that you wouldn't necessarily get from a Shannon information perspective. Finally, we have this developmental reinforcement learning approach, which we're moving towards. This is where you can develop discrete layers, as we showed in the original developmental mode, which will help agents learn better policy. So we can actually focus on developing discrete layers rather than letting things sort of develop randomly. We can look at the growth of the nervous system as a way to counter potentially large continuous action spaces. So if we grow a nervous system to a certain size, it might help the vehicle deal with very large spaces where there's a lot of stuff going on, not just simple stimuli. And then we can actually implement curriculum learning, which is where you have easy examples and then progressively harder examples of some stimulus, and the system can keep up in terms of classification and learning. And so all these things we're kind of thinking about in as terms of future directions. And then finally, we also can think not only about connectivity matrices and these discrete cells and connections, but we can think about development as it exists in organisms where you have cell migrations and structures that form that are sort of, so this is the neural cell crest in chicken. So it's forming this morphology, and the morphology is changing shape along with the cells migrating and assuming different roles during development. All of these can be, we can model these in terms of the acceleration of their rate of growth and their number and their position. And so those sorts of things might be another thing to add to this model as well. And so if you're interested in non-neuronal cognition, which is basically where we're using sort of non-neuronal means to characterize cognition, we have a talk for you Thursday at 12:30 UTC. So then this would be the final slide. Thank you for your attention. And I hope by Thursday that we have a word on our submission to Baby Minds. And so if it's a thumbs up, I'm going to put a accepted at Baby Minds logo here. And then people know where to go to see this in more detail. Thanks to Stefan Z and Ankit for their work on the developmental Bradenberg vehicles. Visit our lab and website, and then a shout out to this group. And that's that talk. So do we have any, what are our thoughts on that?
It's really interesting. I appreciate the overview, and I think that it all makes sense. It's clear.
Yeah.
It was really nice, but I think would it exceed the time limit? It was very clear. It was nice, but I guess it was wasn't it long for the talk?
Yeah,
it take more than 15 minutes.
I'll have to think about how to shorten it up a bit. But I mean, that's just going to be a matter of I'm talking, I'm kind of going stream of consciousness now. I got to tighten it. I mean, because the slides are, that's like the walls, when you have the walls of text, they're kind of they take a while to unpack, but I think I'm going to make some, especially some of the points about the contingency analysis, I'm going to try to summarize those more quickly. So if I can summarize them more quickly, it cuts the time. And then maybe some of the stuff will have to be skipped over for the main talk. That's maybe something people can suggest. What would be what we could skip over if we need to. So yeah, I think that's a consideration is the time factor. So.
Yeah, but I think that if we go 1 or 2 minutes beyond the time, it won't make any difference. There will be that much lenient. But yeah, we can check that if we can make it shorter.
Yeah.
So we all can go through the slides and see.
Okay. Yeah.
Yeah.
Jesse. Oh, you're muted.
Yeah. Nothing outside of what's been said. But yeah, I look forward to that. Let me know, like, I don't know how. It sounds like you'll be just basically be presenting it yourself. I mean, I can probably just attend. I'll be there one way or the other, but let me know about any of that stuff. But I like it overall.
Yeah. I mean, if you want to present part of it, you're welcome to. I didn't really. I don't know how they do that though. I mean, I guess if you're there, you can.
Yeah, it's okay. I'll look. I'll try to figure out a little more about that on there because I hope to be learning more about the process of presenting like this weekend. But either way is fine.
Okay. Yeah. So yeah, I'll put those in the Slack channel, and then Krishna, I can send you the links because you're not in that Slack.
Yeah.
So, so the other one I want to show is the non-neuronal cognition talk. So this is the Psychophysics of Non-Neuronal Cognition. This one has a couple of videos that I was able to process last night. So let's see. So this is based on this organism called Bacillary, which is a type of diatom, and I'll explain what those are. But this is its free movement here in the wild. So you can see that there are these cell colonies that move around. You can see each of these colonies are moving back and forth. They have like these interesting kinds of movements. And then you have a single colony isolated. You can see that they move like an accordion. They go back and forth. So this cell on the leading edge here is moving back and forth, and it's pulling the other cells along with it. And so this cell is actually remaining stationary. So what is this all about? Why do we care? Why do we call it psychophysics? And why do we call it non-neuronal cognition? So we have this, we talk about psychophysics. So psychophysics, we might think of it in terms of humans, but it's actually broad. People have studied this in animal models, in Stebbins and in Sarah's book, in mouse and goldfish and other organisms. But can we also use this method for looking at diatom behavior? That's a good question. That's what we're going to talk about here. So what are diatoms? Diatoms are an algae. So they're actually a eukaryote, but their cells have plant-like characteristics. So they have a chlorophyll element to them. And they have a silica cell wall. So silica is an element that's very hard. And so there's some more reading on diatoms here at the bottom. This is a rapid diatom, which is the type of diatom we're going to be interested in this talk. And this is a single cell. It has a bunch of things going on here. It has like valves and a frustule in the outside of the cell. And that'll become important in a little bit. They're all different shapes of these type of rapid diatoms, so they can acquire different types of shapes. They're only 10 to 80 microns in size, so they're very small, the single cells. This is an analysis of bacillary movement. So if we take that video we saw on the first slide and look at it in more detail, you can see that there's this accordion-like movement. It's a series of oscillators because they move back and forth like this. And that's an idealized movement. So we're kind of interested in the idealized movement for now. There are phases of movement. So there's free sliding. There are all sorts of changes in momentum, as you can see. There's a sort of freezing up. The cells sort of align up here in part of the phase as they're moving back and forth, but they eventually extend all the way out and back. So there are all sorts of phases of movement to understand. And this can be modeled as a series of coupled oscillators, but the movement can vary maybe due to the stimuli that the cell colony is experiencing. So that's why we're interested in the psychophysics aspect. So this isn't just coming out of nowhere. We can actually think about the this sort of a cognition of non-neuronal systems or cell colonies. So the cognitive cell, for example, it's a paper that was published a couple years ago. They define non-neuronal cognition as the total set of mechanisms and processes that underlie information acquisition, storage, processing, utilization. So this is like basically cognition, but in something that isn't a set of brain cells. In this Baluska and Levin paper, they identify the extracellular matrix as ideal as being as acting as a memory medium. This internal stigmergy, as they call it, are physical traces that can be referenced and read in the future. And so in diatoms, we're lucky because we have these. These are mucilage trails that exist in diatoms, and these are sort of connections or secreted polymers that result from these cells moving against one another. So as you saw the cells moving against one another, you notice that they didn't separate, although they're moving constantly. And the reason is because they have these secreted polymers at the interface between the cells that are responsible for adhesion and movements and controlled movements. And so we can actually think of them as some sort of cognitive mechanism. And so diatoms are found in a bunch of different habitats. They're usually aquatic or, like a estuary or, in shallow waters. And so this is the world that they experience. And so there are a number of things that they get in terms of stimuli. One of them is CO2. So they have elevated CO2 levels. So they can sense CO2, they can sense concentrations. This affects the growth of the cells. High CO2 results in reduced grazing pressure. And so there are all these things that CO2 controls. And so it makes sense that they may respond to CO2. They may also respond to different types of variations in the water column. So when you have fluid motion, such as water shear or osmotic stress or iron abundance in the water, then those can all affect the diatom as well. And so they have sensory systems for detecting and responding to these pressures. And finally, CO2 combined with light availability affect diatom physiology. So light intensity has another effect on it. And this is a light source here. So low light affects physiology particularly. And they can also localize sources of chemokinetic and chemotactic mobility. So this light affects the way they move. And so these are all important points from the literature. The idea here is to think about the behavior of these cell colonies with respect to its environment, and then what the pattern responses might be behaviorally. So this is something we propose called a collective pattern generator. And so this is a way to think about these cell colonies more in terms of a brain or a cognitive mechanism. And so you can see that the cell, this type of diatom, moves against the substrate in different ways. They call this gliding locomotion. And it's controlled by an actin-myosin system. So actin filaments and myosin elements are controlling movement against the substratum. And the substratum, in this case, is another cell. So we're looking at two cells moving against one another. But they move in this way, and it's neuronal in the sense that it's muscle. But the muscle isn't really connected to anything neuronal. It's just kind of moving on its own. And so, maybe I'll put a GIF in there, maybe not. This actin and myosin system interacts between cell bodies and produces these collective movements. And so we will build our model around propulsion as the mode of movement. There's a psychophysics model that we want to achieve, which is taking environmental inputs and looking at the actin filament outputs and seeing what they look like. This oscillatory movement that we're going to model is a small subset of the possible behaviors, but it allows us to sort of view the COPG circuit. And psychophysics models we propose accounts for a wide range of behaviors outside of these oscillatory movements as well. So there's some value in building these models outside of that. So this is a CPG. This is a central pattern generator in like a organ, a mammal or something like that. And you can see that they're inhibitory synapses and excitatory synapses. So it's a collection of neurons that are connected in different ways, and it produces this patterned output that's cyclical. So it's like a walking or some other, like a heartbeat. Those are all CPG mediated or controlled things. The COPG, by contrast, is these cells that are sliding against one another. We can think of this as loci of sort of actin-myosin activation, and then they're connected by these imaginary connections that are weighted. So we can think of this in terms of like different points along the cell bodies and how they're related to one another as they're moving. So the speed of the movement and the relative position of the cell, those points in the cell, are all sort of part of this model, where we can look at the weights, and they're weighted by their, their movement, their position relative to one another, and so forth. And so we predict that they behave much like these CPGs in terms of generating a rhythmic pattern of behavior. We can also use a series of coupled sinusoids to model an ideal COPG. So we can use this type of approach where each cell is represented by a sinusoid. And these sinusoids overlap at a certain phase. And so it could be a quarter phase, it could be half phase, and so forth. And this is an idealized model. But as you saw in the movement videos, this doesn't necessarily fully describe the movement. So we can do things like we can add additive noise to the signal. We can adjust the sort of the sinusoid phase. We can do a lot of things with this type of modeling. And so the speed of the movement, the derivatives of position can also be varied according to the effects of the stimulus. So we can model this, and we can think about the stimulus as sort of deforming this idealized model, either through noise or through speed ups and slowdowns in movement, things like that. Then this is the potential psychophysical measures. So I could go through this, I'll probably walk through this with some examples. Signal to noise ratio, sensory thresholds, and differencing between the stretch phase. And that'll just kind of draw from both of these slides. So that's not done yet, but that would be the last slide. And then thank you for your attention. And the data are Thomas Harbeck, who's a collaborator in Germany, is doing a lot of the observations of diatoms. And then DevaWorm Group is kind of looking at this, thinking about it in a new way. And so that's the presentation for that. Any comments or questions on that?
It's the same thing that Dick was talking about.
Yeah.
Yeah, he was talking about that jerk and that diagram movement, I guess.
Yeah. This is interpreting it in a little bit different way, but this is one of these things where we're proposing this as a new way to look at it, and so most of the people at this conference won't be familiar with this model organism, but it's that's why you have to describe it a little bit beforehand. But yeah, it's like describing the motion. Why does it move the way it does? There's so much we don't know about, like, exactly why. So we don't really know why the brain does what it does relative to a stimulus. So this is not like a new problem in this community, but it's, Jesse or Anson, do you have anything to?
Yeah. You can add. I've kind of seen it a few times, so, but I think it'll be a good presentation.
Yeah.
I don't have much to add, but I found it interesting. The way you explained it was really coherent.
Okay. Yeah. Thanks. So that's the two talks that we have, group talks, and we have other talks as well. So hopefully this, it's a successful week. I know we also talked about doing a blog post. That's something that's still up in the air. I mean, it's like we have a lot of stuff this week, so if we don't get to it, it's okay. But it's there. We'll probably have like another week after that where people will be thinking about it. So, and then this coming week, I hope to hear back about the Baby Minds paper. They were supposed to last on Friday, but I think they moved back a couple of days because of they probably were busy things. So hopefully we'll hear about that by the time this talk gets presented on Thursday. And that's a lot of that. Some of that is drawn from that paper, so that's why it would be nice to have like that, be able like have it available, or like have a stamp on it that says it's on its way out. But that's good, and that would tie in. Then people would know where to go next to see more work on it. In terms of like the Baby Minds paper, I think if it gets accepted, we have a little bit of time to turn it around, and then they want us to submit it again with revisions, and then we can actually make it available on as a preprint, so that people can actually read it publicly, and that'll be a nice tie-in to and then we can maybe like take this slide deck and the Baby Minds paper and promote it a little bit on social media prior to the workshop, and then the workshop will happen, and that'll be a good way to promote that kind of work. So I want to actually also, now, are there any more comments on the Neuromatch stuff?
I think
Okay. So I wanted to talk about one more thing today, I guess. And this is that Jesse has been setting up. I've been talking to Jesse and Rashab about this, but Jesse actually setting up this new GitHub thing. Do you want to present that, or do you want me to share the screen on that, Jesse?
I will share my screen. Let me see if I can do this correctly here. I'll get to choose here. Okay. There. So yes, this is the organization page. I don't really know how to really call it, but it's in GitHub. Like as you may recall, like if you look over here, I think you can see this right. Yeah, this is like what we're all familiar with, and it's a great repo, sort of our, I don't know, legacy home base stuff. Most of the updates for things will be here for a while. Like this is our typical one. But I've come through and I've been experimenting with a lot of things on the organizational aspect in GitHub recently, and just to kind of put this here, we have it here, and I'm slowly transferring over some of the more refined or finished products. Like we did set up the epistemological directors one, which is essentially just the link to other things in the old repo, the traditional repo, taking off of this is what the directory's front page, front end was there too. And we're going to do this with some of the other repos too. Like I think, I forgot what the ones we talked about. Now developmental brain vehicles is one of the big ones. Are we going to do contextual geometric structures too?
Yeah, I think so.
Okay. And I think there was one other one that I'm forgetting currently.
I think probably like the proposals for different things.
Oh yeah. Okay. I'll talk to you more about that because that was sort of a Yeah. But with repos, and I like it's a good way to manage a lot of stuff with people here. Like you're seeing sort of the admin view, but like it's nice to have teams. You can break people on different projects, and it will always we always have issues about get someone write access to this thing when they want to actually start writing code to it, or manage their pull requests or whatever else. And this will help it a little bit. And as you can see, we've got like Ankit and Stefan have joined the lab, and they're kind of in our GSOC team, and eventually that's also tied to like I've associated that with this repo, our DVD repo. And as like I was talking about with Bradly yesterday, eventually we're going to put some specific, we'll put specific instances of the software that was made for that project here and other projects as well. So in short, we're kind of using, for the time being, this lab, this page, the organizations page, will be a way to host either some finished, refined, actual coding projects and presentations, and some of the making it a little bit easier to navigate the many different repos and existing stuff that's been in the legacy traditional account, I guess. And then also slowly developing new things in these areas, and trying to sort of take an opportunity to flesh out some of the bigger I don't know, silos or collections of things that we're doing in the lab. So yeah, it's mostly just kind of back-end data management stuff, but I think it'll be a nice way. A lot of times people come to the lab and check out the lab and like, oh, what are you doing? What's project? What we're working on? And I think this would be a helpful management way that bridges. You can say, oh well. Here are some teams, like here's the Neuromatch team. I don't have anything in here yet. Actually, I do have everything here. This is actually what I was talking about. I forgot. I gave up the update for a couple weeks ago. I did this. This is basically a post I made in Slack, but I put it here with other people doing, their things. And it's great that we're having so many things in Neuromatch, and also hopefully NeuroIPS too. But this is what I was talking about at the very start of the meeting too, regarding looking for hosts or MCs or backend help on the presentations. So yeah, check out the Airtable link here for anyone. But like it's cool because you have a little bit of a hub of discussion around a topic. You can easily go to the members, the teams, and the repos. Like you can just go. You can find, like, this could be a very accessible, and it doesn't have to be like, it's not going to be, obviously this is much more public facing and whatnot than our Slack. Our Slack will be private discussion and the nuts and bolts, but you can put major updates here. It's sort of like, it kind of feels like, if you're familiar with ResearchGate and how ResearchGate is, like, it's almost like a step towards that of being a little more public, being a little more transparent, a little more open. And I don't feel like this is going to be, like, this will not be actively updated in the way that, Slack is. We'll constantly have discussion there, or I'm not interpreting GitHub as social media either. I'm not trying to go in that direction, but I think having a hub to show highlights, or like similar to ResearchGate, you show updates on projects, and having that specific to project development, or breakthroughs or milestones in the code we're working on or presentations that we're making. Like we could eventually put some of the things that we finish up here for Neuromatch 3 and put them in this section. Like I'll make a repo that's specific to Neuromatch 3, and we could put all lab materials in here and just have a very easy way to show, hey, what did okay, well, you guys were at Neuromatch 3, what did you do? Okay, boom, 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 boom. And I think it's not that we don't have that capacity in our old lab, but I think it's be a much more accessible way to show it here. And also, some of these aren't, a lot of these are in-house things, but especially for events or things that are more open, or we're not really participating in Hacktoberfest, but things that like the Neuromatch Academy, where a lot of people came into the lab, some of the hackathons and events that we participate in that are very outward facing. I think that will also be a much easier way to quickly direct people, because a lot of times people come into the lab and be like, oh, what's going on? We say things, come to a meeting, or I point them to Slack channels, and it's sort of like, it kind of is, you can be lost in a bit of a maze. And I think between this and using it sort of a auxiliary tool or map, if you will, in association with the websites that we have for the different sort of project groups, I think there's a potential to have a coherent web presence. And yes, it's kind of using GitHub as in a project management sense, but I think there's a lot of opportunity to really blend those things nicely, and continue and show. It'll be a nice way to, there may be some experimentation with what to use in this versus our legacy. Like here's the MetaBrain models one in our legacy repo, and we don't, obviously I don't think we're gonna be transferring any repos at this point. Well, we might have some backwards linking things like we're doing for this this dashboard. All these links actually go back to the legacy repo, and I think that's very fine for certain things. But I don't really see an immediate need to transfer some of these things. But I think making the new hub for Neuromatch stuff, having all that in one space, I think that'll be very good. Having a lot of the Google Summer of Code stuff in one space will be pretty good. And having a macro sense of, okay, well, what each year, we've been participating in this either directly or indirectly, and what have we been doing? I think being able to see that cumulative growth in this format would be very useful and good. And other things too, like the MetaBrain models, and I'm not quite sure yet how to slice up MetaBrain models versus, I made a team here that was, one of the teams is representational brains and phenotypes, and that's sort of like a little bit of echo here. Representational brains and phenotypes. Were you going to say something, Krishna? That I might mute. Anyway, sorry. There we go. Okay. Oh, hold on. the screen. Yeah, representational brain and phenotypes is like one of the major like umbrella research teams in the whole lab. So I'm not sure quite yet what to do with that, but I think, yeah, I think there'll be some specific repos for MetaBrain 1, the MetaBrain group, and then also I know we just talked about adding proposals back to this. So that's pretty much it. If you have any comments or questions, also make sure you check your go-to. I believe it's here in notifications. There should be. I've invited pretty much everybody that I could find. So go to your notifications and accept invitation to the lab. I've also set one up for my own subgroup as well.
I looked at my notifications and I didn't see anything.
I don't know what to say. This happens sometimes, but like you're invited. I can resend the invitation. Again, know that you're here. I think that's you, right?
Anson Lim?
Yeah.
Huh.
I'll take the invitation and see what happens. Anyway. So yeah, I've set up this one for my own, like I'll be doing a lot more here as time comes, but I'm kind of transferring. This is the old cognition study group main interface with things to be built out here. Pre-existing introduction, I'll be doing that later today. But there's short version, there'll be things to come. And if I don't know. I mean, if anybody else has been getting the invitations, do you see them in your notifications, or where do you see them?
I got them in my email. Like it
Oh,
links through to the Actually, I don't think I saw in the notifications. I don't remember. So I just used an email link.
Okay, well, maybe check your email then, I guess.
Yeah, check your spam folder and yeah.
Yeah. That's pretty much it for. I mean, any questions or comments about GitHub stuff?
Yeah, I had a few. So this is what do they call this? Is this a organization, or you said it wasn't an organization technically? It was a team, or
This is the organization page in GitHub.
okay.
Is the new one that we made.
Okay.
And there are teams within the organization that you can break out, and you can make people, you have repos, and you have projects which are basically you can make the you know. I think there's potential here for in lab managers, like, you know, organizational idealized dreamland, whatever, would create an actual. There's one major task board that can aggregate smaller ones within it, which is sort of what I've done in some personal projects. But that's I haven't had any time to do that. Like I've done some stuff here this week, but obviously, you know, this isn't really the biggest priority right now. So there will be more to come. I'll probably have some updates on this in the coming weeks as well. So
Yeah, I like the way this is set up. I see I know we talked about these yesterday, but like the some of these epistemological directories is like an amalgamation of everything that was scattered all over the place. That's why that's very useful. So we might work on this a bit. I don't know what we would put in this repo. Maybe like, because we have different instances basically that we're pointing to, but then there's probably something to add to this. Now, this is something that is this is basically a repo that's been copied over from the old repo, but we don't want to do that in every case because we have a lot of legacy things like forks and stars and links and papers and things like that. So that's not. But this is a good summary. So I mean, I think like a front-facing thing for projects, like if you do one for CGS, we might have it where it's like a README, and then it has maybe a list of resources that are now scattered all over the place, like the GSOC projects, and maybe some of the other like that, something like that, with a little bit more description.
Repo. Yeah, this is the current CGS repo, and there's like some things here, but I think making it new, because CGS has been a significant part of like a lot of discussions, and it even sort of like was, I mean, that's a big part of my last Google Summer Code proposal. So it's a major theme in the lab, and it has its own, one thing about repos in a new group is sort of like, it's sort of the sense of what are the biggest, like very much more, I guess, coarse, as opposed to a lot of the granular stuff that's been here in the old one. And it's not, I think there's a way to do it. There's a way to do it in a there's a decent way to do things where our old traditional account, I guess that's what it really should be called. This one here, Orthogonal Research Lab. I guess we'll call that a traditional account or old account or whatever. Like, that's a place where you can pretty much throw whatever you would like to do. And I feel like putting it's not quite the same as like beta versus production, but putting it more towards a public facing, bigger updates on this page, I think will be good. And we want to like bring in, I don't want to lose at all or like not show things. But I think there are ways to, like, when we bring over the contextual geometric structures, like, yes, I'll link to these things and like these are valuable things as well. But there's also, I think there'll be ways to facilitate, and even potentially creating, like I haven't looked at these set of issues here, but like these issues are important. We don't want to lose those, but ways to also like I don't know.
Yeah.
Ways to facilitate new development in old areas too. Put it like that.
Yeah. Well, I think this is good, a very good start, and I would we'll talk about this more in subsequent weeks. So let's thanks for making everyone aware of what's there now. And if you are not joined as a member of that, check your email for an invite. If you didn't get an invite, let us know on Slack, Jesse or myself. We can figure out how to get you in there, and then join a team, because that might be a good way to communicate to team members for things that are more formal teams. Because in Slack, we have the channels, and it's hard to, sometimes there's a lot of drift in the channels in terms of discussion. So this would be a good formal team exercise. Yeah. And I'm glad this is actually up before Neuromatch, because now we can, people get interested during Neuromatch. It's now a new mechanism to get them into the mix.
Yeah, and like what will be really good about this is you can set up a lot of, like, I probably should have said this first, number one. A lot of people aren't going to be GitHub heavy users, and that's fine. I'm not expecting that or demanding that or anything of that nature. But it'd be good to join the group just to show that you're there and show on GitHub that you're active in that sense. But like, I definitely encourage people to use it. But I think it's also, it will be a very good way to have people who, because you can set up different roles, like you can set up an outside collaborator or people who are no longer directly working with a lab, or people who are sort of like, not really affiliated with us but want to work on things. It kind of lends, it doesn't, again, it's not quite, it's not a social network, and it's not quite linking, but it's still GitHub, and GitHub has a very sort of, it's I don't know what to really describe it as. It's sort of like a LinkedIn and a social network for code and for project management around code and development and sort of things. So having a presence there and having something structured like this, I think, has a lot of important things. And yes, for Neuromatch too.
Yeah.
So yes, that's. It'll be good to flesh that out over time. It's kind of a side project, and obviously I have other things to work on now, but it came up because I'm fleshing out a lot of things that I'm doing for my own presentation are adjacent and tied to my development with my own. My group here.
Yeah.
So it's sort of like I've been experimenting with that, and I was like, I might as well do the orthogonal stuff at the same time, which is why we're here. And like, yes, this looks good. And so I'm learning to do this, I'm learning to do that, and it's coming along nicely. So yeah.
Good.
Okay.
Yeah.
Last thing I wanted to quickly mention is this NeuroNovember event. One of my friends is sort of trying to host it. Project Encephalon and Stimulus. Registration's opening soon. Submissions are open. It's basically a neuroscience open-ended. I think they're even having some like hackathon kind of things at it. But yeah, check it out. NeuroNovember. But yeah, there's NeuroArt, NeuroFiction, 2-minute solution, poster presentations, corner science talks. And I think this is sort of much better too. It's it's kind of an up and coming thing. But check it out if you want more to do after Neuromatch. There you go. That's my plug for them.
All right.
But that's it. I think there's pretty cool meetings. So unless there's anything else, for me.
Yeah, it looks good. Yeah, thanks for sharing. All right. So, Anson, do you have anything else? Any last words?
Not as of right now. I'm gonna work with Justin to get added to the new page, and besides that, I do have by next week. I should be able to talk about the AI-related project because this Friday's the deadline for the project submission.
Yeah.
So
yeah, I think we'll be working on the trajectory thing a bit, and then I look forward to that. I think that'll be a fun. I look forward to hearing about that next weekend for sure.
Yeah. I look forward to it. So,
yeah, same here.
Yeah. Well, thanks for meeting. If you're watching us online, check out the new GitHub organization, and let's keep in contact about the presentations if you're giving a presentation. Otherwise, see you at your assigned time, and hope this week goes well for everyone.
Yeah.
Okay. Yeah.
Congratulations to everybody who's like. There's a lot of presentations going on, and a lot of people are really busy right now. And I've talked to not everybody's here in this meeting, but like I've been seeing people busy and hearing them what they're doing, and it's an exciting time. It's a busy time. Like I have I'm like in graduate school application stuff as well, so it's just like it's a really busy fall, and like college football is back, which is a great thing for me because I don't know if it's going to happen all this season. So I'm going to go watch college football for a very little bit and do writing presentations the rest of the weekend and applications. And it's a busy time in a lot of ways, but I think everybody should be pretty excited and good job for what's going on and a lot happening around. So yeah.
Okay. Thanks for attending. See you next week, or see you this week during the Neuromatch.
All right, bye.